Hello, good morning, good afternoon, and welcome. We're going to take just a quick moment here to let everyone join in and get settled, and we'll begin in just a few moments. All right, well, hello everyone, and welcome to today's webinar titled Comprehensive Profiling of 200 Plus Proteins Using the NULISAseq Inflammation Panel Reveals Unique Signatures of Immune Response in AML. This webinar is a part of the 11th annual event in the Cancer Research and Immuno-Oncology Week virtual event series. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Alamar Biosciences. For more information about Alamar Biosciences, go to www.alamarbio.com. Before we begin, I would like to remind everyone that this event is interactive, and we encourage you to participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the event. All right, now let's get started. I now present today's speakers, Alex Forrest-Hay, VP of Sales at Alamar Biosciences, and Dr. Hussein Abbas, MD Anderson Cancer Center. For a complete biography on our speakers, please find it on the left of your screen. Alex, I'll hand it over to you to get us started.
Thank you very much for the introduction and the opportunity to talk today. My name is Alex Forrest-Hay. I'm the VP of Sales at Alamar Biosciences. Today, I'm going to talk to you about a groundbreaking new proteomics technology called NULISA, which really is an ultra-sensitive liquid biopsy proteomics platform developed by Alamar Biosciences to enable ultra-high sensitivity detection of protein biomarkers from liquid samples. So a brief introduction to Alamar Biosciences. The company was actually founded in 2018 by the same team of scientists that originally founded a company called Advanced Cell Diagnostics and developed the RNA-scope technology for highly sensitive in situ RNA analysis that became one of the founding spatial biology companies. So the founders founded Alamar to turn their attentions to proteins with the real goal of powering precision and ultra-sensitive proteomic detection for early detection of disease, in particular, early detection of cancer. So Alamar has actually patented and invented three core technologies to enable ultra-high sensitivity proteomics. The first is a technology called NULISA, nucleic acid-linked immunosandwich assay. And this is an ultra-sensitive, attomolar level, NGS-enabled digital immunoassay platform for protein analysis from either single biomarkers up to highly multiplexed panels. We also have a technology called Attobody, and have recently spun out a company called Attavia, which is focused on using antibodies for developing therapeutic antibodies. But antibodies can also be used for developing NULISA assays with ultra-high sensitivity. And these biparatopic nanobodies have extremely high specificity and sensitivity. And the third technology, which is very important, that we've invented, is the ARGO instrumentation. And this is for the first time now a fully automated proteomics platform, which enables you to take very small amounts of sample and with a fully automated workflow, generate data very quickly with high reproducibility. So really the goal here and the ultimate goal for Alamar is to provide automated solutions for ultra-sensitive protein analysis across a range of multiplex levels to detect critical biomarkers and really to be able to go from discovery right through to the clinic. So at the highest level, NULISA is the most sensitive high-contact immunoassay fluid biomarker platform. So we really have unprecedented sensitivity, and I'll show you some data to support that claim. We're able to detect protein biomarkers at the femtogram per mil level, and this is extremely important when you're thinking about early detection of disease or monitoring patient response. Being able to measure these proteins at this really low level, A, gives you the ability to measure them in baseline healthy samples but also at the earliest stages of disease or response to therapy. So we can now quantify very low abundant proteins utilizing NULISA. We also have a very wide dynamic range, greater than 10 logs of dynamic range within any given assay across the multiplexed panels. And this allows us to quantify proteins at a very wide range of biological expression levels. And we don't have to dilute samples, which makes it very easy to generate the data. And we have a very flexible chemistry. So as I said, we can measure single biomarkers with very high sensitivity. And we can also develop panels of hundreds or potentially in the future thousands of targets from a single experiment. And we've already tested this technology in a wide range of sample types. And I'll show you some data today from blood, serum, dried blood spots, and also CSF. And the ARGO instrument enables full automation. So really very easy to set up the system, minimal hands-on time, single technician can generate this data in a highly reproducible way. So coupled together, this makes this platform, the NULISA platform, the most sensitive and high throughput proteomics platform available. So the key thing here is really enabling a new level of sensitivity with high-plex proteomics. And this is absolutely critical to enable detection of known biomarkers at lower levels, to enable the discovery of novel biomarkers that are currently undetectable, and to be able to detect biomarkers at the earliest stage of disease progression for early detection. And so by having this level of sensitivity, we can now understand a clearer definition of what a healthy baseline looks like, and then ultimately look at the small changes that are occurring earlier in disease or predict biomarkers that can predict therapeutic response. So how does NULISA achieve this incredible sensitivity? So we have a proprietary technology that essentially removes background from the matrix and from the sample before we amplify the signal and measure it using nucleic acid readouts. So here in this diagram, you can see two antibodies, the green and the yellow antibody. And both of these antibodies are required to bind to the protein of interest to create a signal. And those antibodies bind to two different epitopes. So this itself gives very high specificity. To these antibodies, we conjugate oligonucleotide sequences with a known barcode. And so we can then leverage these sequences by either next generation sequencing or qPCR to generate the signal and measure the protein quantities. What makes NULISA so sensitive and so specific is that we have this poly-A tail conjugated to one of the oligos, so one of the antibodies, and we have a biotinylated oligo conjugated to the second antibody. And we utilize these sequences to essentially capture the immune complex onto paramagnetic beads, and then we remove background with sequential capture and wash steps, which gives us the ability to improve sensitivity by about 10,000-fold over traditional approaches. So this is essentially an evolution of the proximity ligation assay, where we remove background to improve sensitivity, and it creates a highly specific, highly sensitive platform. Just to go through the steps of the workflow, step one is the same as any conventional immunoassay. So here we incubate the antibody pairs into the solution, and we typically use 10 microliters of blood or plasma or serum, and we do a 2-hour incubation. So this enables the antibodies to bind to the protein of interest and essentially form this immune complex with two oligo sequences in close proximity to each other. The next step of the assay is where NULISA becomes unique. So we introduce oligo-DT beads into the sample, and this enables us to capture the immune complex using the poly-A tail on the first antibody, binding to the oligo-DT on the beads. So once it's captured, we can then actually remove background with the wash step. So here we're removing all pre-detection antibodies, analytes, and matrix, but we're retaining a specific analyte bound by the capture antibody. So that's the first capture and wash step. We then release this back into solution, and we then, as a fifth step, introduce streptavidin beads. And this really utilizes the biotin on the second antibody to capture the immune complex for a second time. And this enables us to do a second wash step, which then removes all antibodies and residual matrix, not complexed with the analyte and the second detection antibody. So by the time we've done these steps, we've essentially purified the immune complex and removed all sources of potential background to drastically improve the signal-to-noise ratio. So the next step is a ligation step to join the two proximal ends of the oligos together. So we introduce an overlapping sequence that overlaps the two ends and then ligate them together. And once that ligation step is complete, we can then measure the proteins via either qPCR for our singleplex assays or by using an Illumina NGS readout for the multiplexed assays. So this really leverages the power of nucleic acid detection as a proxy for measuring the protein concentration. So this slide shows the advantage of the sequential capture and wash steps of the NULISA assay. In this case, we took the same antibody pairs for IL-4, an important immunological biomarker, and we performed an analysis with the typical proximity ligation assay and with NULISA. And what you can see from this diagram here is that we have a 10,000-fold improvement in the limit of detection with the NULISA assay versus the typical proximity ligation assay. And we also have a much broader dynamic range across 7 or almost 8 logs in this case for IL-4. So what this really enables us to do is now measure IL-4 previously impossible levels and also measure it across a very wide, broad dynamic range. Here's another example, actually, where we've compared NULISA to the current best-in-class technology from Quanterix called Simoa. Now, Simoa is a singleplex proteomics technology capable of very high sensitivity. And here we're measuring HIV P24 protein with the same antibodies, comparing Simoa and NULISA. And you can see versus Simoa, we have about a 10x improvement in the limit of detection, and again, a broader dynamic range across about 8 logs. And for this particular protein, we're able to measure 0.24 femtogram per mils as a limit of detection. So exceptionally high sensitivity and showing that we're actually improving sensitivity over the current best-in-class technologies. And so if you look at this across a wide range of cytokines and chemokines, which typically are very low levels in blood samples, you can see here in this chart that NULISA can achieve lower limits of detection, lower limits of quantification, and as a consequence of that sensitivity, we can now detect all of these critical biomarkers in 100% of healthy donor plasma samples. And this is absolutely critical if you want to understand the underlying biology and the changes that are occurring early in disease. So this 6 graphs here show that we really are achieving exceptionally high sensitivity for critical markers like IL-4, which we know influences pathways involved in cancer cell survival and metastasis, and LIF, which is a potentially important therapeutic target in pancreatic cancer. So here we're showing really, really high sensitivity, the ability to detect 1.3 femtogram per mil of IL-4 from a human blood sample. So when we develop NULISA assays, we go to great lengths to ensure reproducibility and high performance. So here's an example of IL-1 beta, again, an important biomarker in the production of cancer cells and inflammasome response. And here we can see that NULISA actually is able to measure with a limit of detection 0.003 picograms per mil, which is again almost 5-fold more sensitive than the current best technology Simoa. And what we also do with our assays is we look at parallelism across a wide dilution range to make sure that we're getting a linear response as these protein levels go down. And we also do spike recovery experiments to look at things like matrix effects. And because of these capture and wash steps, what we see is exceptionally high performance and reproducibility for all of these assays, as well as our ultimate goal, which is to have 100% detectability in plasma samples. And here's a nice example of IL-2, which again is another important marker, particularly in metastatic renal cell carcinoma and melanoma. And here you can see again that we have an exceptionally high sensitivity assay, and we're able to detect in 100% of samples IL-2 from healthy blood donors. And here we're comparing it to the mesoscale discovery platform and the Simoa platform, and showing better detectability and higher performance. So these are really important cancer markers that NULISA is able to detect at very high sensitivity and with very high performance. And we can also develop assays for post-translational modifications. This is p-tau217, which is an absolutely critical marker in Alzheimer's disease. And here we're showing best-in-class sensitivity and performance for this very important biomarker. So that's showing data from our singleplex assays. But what's really critical and unique about NULISA is we can achieve these high levels of sensitivity also in a multiplex setting by using the NGS readout. And so here you can see 5 important cytokines where we've actually measured them as singleplex assays with the qPCR readout at the top, and then with the multiplexed NGS readout at the bottom. And these are 5 cytokines from LPS-stimulated whole blood samples from 3 healthy donors. And so what you can see here is essentially very good correlation, the same dynamic range and the same sensitivity for both the singleplex readout and the multiplex readout. And this is really important as it enables us now to build panels of biomarkers which are critical across different disease pathways. So the first panel that we've built, which Dr. Abbas will present some data on after me, is called the NULISAseq 250-plex inflammation panel. And this panel gives the most complete coverage of cytokines and chemokines and growth factors on the market, and also includes several critical, or many critical cancer markers. So with this panel, we're able to measure 250 protein biomarkers from 10 microliters of plasma or serum or a liquid sample. And we can actually measure all of these markers with exceptionally high sensitivity across a very broad dynamic range, about 12 logs of signal that we can measure without any sample dilution requirement. And because of the automated nature of our platform, the reproducibility for these panels is exceptionally high. And to achieve high levels of low CVs with low abundant proteins is very challenging and shows the power of the NULISA platform. I'll also mention that these panels are customizable. So if you want to add markers to these panels or take subsets of these panels and develop custom panels, that's actually quite straightforward to do and part of the NULISA offering as well. So this is a preprint publication that's currently in bioRxiv and in late stage review for publication. And it's really the introduction of the NULISA platform. It's called NULISA, a novel proteomic liquid biopsy platform with attomolar sensitivity and high multiplexing. And I'm going to show you a little bit of data from this paper that really shows the power of this platform and compares it to other current proteomics platforms. So in this paper, we actually showcase data from about 151 samples that were randomized across 2 plates. And these were a mix of healthy donor samples and different disease samples, including cancer samples. And this enabled us to actually look and compare the NULISAseq inflammation panel. In this case, it was a 204-plex panel, which we've subsequently expanded to 250. And we compared it to an Olink Explore panel, which had 368 targets. So across those 2 panels, there were 92 common targets, which we used to assess intraplate CVs, detectability, and also looked at differential expression patterns for the disease groups versus the healthy samples. So I'll go through some of that data now. First of all, when you look at detectability, the NULISAseq platform outperformed both mesostele discovery and Olink Explore in this sample set. There were a couple of markers at the bottom where the detectability for NULISAseq was quite low, and we've subsequently replaced those antibodies and improved that significantly. So what you can see is we have high detectability for all of these critical biomarkers, whereas some of the other platforms, the detectability drops off for some of the lower abundant proteins. When you look at concordance between the platforms, it's actually very good, where the platforms have high detectability, but as soon as detectability starts to drop with one of the platforms, then the concordance drops down accordingly. When you look at the CVs, we've got intraplate and interplate CV data here comparing NULISAseq with the Olink Explore, and you can see we have extremely tight intraplate CVs with a mean of 7.3%, and the interplate CVs with a mean of 11.6%. So this really shows that the vast majority of the markers that we're detecting with high detectability and high sensitivity have CVs much lower than 20%. So the sensitivity of our assay is really critical. And I think this data here really showcases why this is important. So here we compared inflammation disease with healthy donor samples. And in these inflammatory disease patients, we saw a high level of concordance between those markers that were differentially expressed between Olink Explorer and the NULISAseq platform. But there were a whole subset of markers on the left-hand side here where they were only seen to be differentially expressed with the NULISA platform. And when you actually look at these markers, and you can see the lasso here, these are very, very low abundant proteins. And the reason that they were not seen as differentially expressed with the competitive platform was that they were not detectable in the healthy samples. And so here it really shows the value of being able to detect these markers with high sensitivity and to really understand the true underlying biology of what's happening within these samples. And here is some data from the cancer samples that we ran. So you can see we ran this panel across a wide range of different cancers with fairly small n. But what's interesting here is that I think there were 46 upregulated targets and 2 downregulated targets associated with these different cancers. And you can see here the hierarchical clustering, which really shows that there are specific protein signatures associated with all of these different cancers. And you can also see how they vary to the healthy controls. So you can see some very high expressing proteins changing in the cancer samples versus the healthy controls, showing the power of looking at proteomic signatures for potentially monitoring disease response or subtyping different cancer types. And just to show you the volcano plot, when you actually look at this differential protein abundance for cancer in these 32 samples versus the controls, you can see some of the key markers that are popping up here. They're typically very low abundant and difficult to detect, like IL-6, IL-10, LIF, IL-18, for example. And also the interferon molecules, which are really key molecules in inflammatory response to therapies, for example. So this is really showing that within a panel of these 200 markers, we're able to see a very large number of differentially expressed proteins in blood samples that are associated with different cancers. So obviously early detection is an absolutely critical area where liquid biopsies can be extremely powerful. And we believe at Alamar that proteins can actually really be exceptionally powerful for early detection of disease. Here is just a list of some of the key early detection biomarkers where we've developed ultra high sensitive NULISA assays. And you can see these are across a very wide range of different cancers. And we're able to put these into a single panel to develop an early detection cancer panel that would be highly sensitive and highly specific. And I'm going to show a little bit of data now for a particular marker that we've developed in collaboration with a leading cancer center called Line1, which is actually a multi-cancer biomarker. So Line1 was recently published in a paper to show that it's actually extremely overexpressed in many different cancers. Typically 2/3 of human cancers show striking levels of the ORF1p Line1 protein. And it's generally at very low levels in healthy samples. So this means that this particular marker could have a very high utility for detecting cancer very early. But clearly sensitivity is going to be absolutely critical when measuring Line1 ORF1p and making it useful as a pan-cancer biomarker. So we developed a NULISA assay for Line1, which has the capability of detection of about 4 femtogram per mil. And that means that we're now able to detect Line1 with 100% detectability in human plasma samples. And here on the right-hand side, just showcasing a little bit of data in colorectal cancer samples, where we're showing a clear increase in Line1 based on the stage of these colorectal cancer samples. And this slide just shows that this is across multiple cancers where Line1 is upregulated. And what's interesting about this is from a paper from the publication, is that some of these healthy samples that actually have high levels of Line1 actually weren't healthy and turned out to have early stage cancer. What you can see here is for cancers like ovarian cancer, colorectal cancer, and breast cancer, there are significantly elevated levels of Line1, but having high sensitivity will enable us to detect these changes in Line1 expression at much earlier stage of disease. And this is a really important differentiator for NULISA, with it having the high sensitivity detection of protein biomarkers. So just to wrap up this presentation, I wanted to introduce the ARGO instrument, because this is really a game-changing instrument because it takes all of the steps of the NULISA assay and completely automates the process. So here we provide the consumables and the instrumentation to enable you to run 3 96-well plates per day with a single technician. And really, we're also providing software to enable QC and also statistical analysis of the data to enable high throughput generation of NULISA data with very minimal hands-on time. And so the ARGO instrument is a benchtop instrument. It takes less than 30 minutes to set up a run. For the singleplex assays, you can generate data with an onboard qPCR system in about 6 hours. For the multiplexed assays, the output from the ARGO is actually an NGS-ready library, which then, in a single tube, gets loaded onto an Illumina NextSeq sequencer for the analysis. So the total runtime is about 14 hours, from 6 hours on the ARGO and then 8 hours on the Illumina sequencer. And that enables you to generate data from 88 samples across hundreds of biomarkers. And this is a high throughput system. So a single technician can load 3 plates, complete walkaway automation, and generate high quality, high reproducible data with very few hands-on steps. And we have this integrated data analysis system, which is actually cloud-based. So the data gets delivered straight to your laptop or PC, so you can do the analysis downstream and get to the biological insights quickly. So the final slide here is just to show that right now we also have a technology access program, which is run through our service lab in Fremont, California. And this enables you to generate data with NULISA on samples that you wish to send out. And so we can generate data on our panels, either the inflammation panel, which I showed you earlier, or the 120-plex CNS disease panel. And we run 88 samples per plate and typically utilize 10 microliters of sample input, although request 50 microliters for the service lab. And we can test this in multiple different sample types, including cell culture media, urine, CSF, plasma, and serum. And the readout for the multiplex panels comes off of the Illumina sequencer, and we typically turn these projects around in 2 to 3 weeks and provide bioinformatics support as well. And then we also have these individual singleplex assays with ultra high sensitivity and can also develop custom biomarkers via our technology access program. For the singleplex assays, we run 74 samples per plate. Typically, that's 37 samples in duplicate. This provides absolute measurements of these protein biomarkers with high sensitivity. And again, the typical turnaround time is 2 to 3 weeks. So if you're interested in sending samples to Alamar, we can discuss that. And so thank you very much for your attention. I hope this presentation was of interest. My contact details are here. My name is Alex Forrest-Hay, and my email address and phone number are there. And you can also access our publication by clicking on this link. And so now I'll hand over to Dr. Abbas from MD Anderson Cancer Center, who's been utilizing the NULISA platform for some of his leukemia research. Thanks for listening.
All right, well, thank you, Alex, for the introduction and for the opportunity to be here today and present some of this work and the application of NULISAseq in acute myeloid leukemia. So the title of my talk is about multiplexing cytokine profiling in AML as a discovery approach, but also potentially as something that could help us understand better what's happening with the patients at the clinical side. So acute myeloid leukemia is a cancer of the blood. It occurs in the bone marrow, and it is associated with differentiation arrest. There's a rapid increase in cell counts that occurs that patients become acutely ill. And this is something I'm going to come back to in a bit to try to understand why these patients sometimes become very ill. If it's not just related to anemia, what other features make them this sick? The treatment is generally related to intensive chemotherapy with a lot of side effects kind of affecting quality of life. And unfortunately, there's a high relapse and mortality rates that occur. At the same time, we need to think about the AML growing in the bone marrow where their immune cells also exist. And that means there's a lot of interaction that could happen between an AML cell and the immune cell that could lead to some phenotypes such as inflammation, which are worth also noting in that context. And they could also impact potentially outcomes. So the diagnostic workup of a leukemia starts with a bone marrow biopsy. Requires also a lot of histopathologic diagnosis, working with hematopathologists, looking at the cytogenetics and karyotyping, a lot of mutation profiling, a lot of immunophenotyping. And not surprisingly, those immunophenotype, if you think about them, they're actually myeloid cells, meaning they're basically antigen-presenting cells with an underlying inflammatory component that exists at baseline by the virtue of them being hematopoietic myeloid cells. And that could have implications as well about their interactions with what happens in the microenvironment. So our lab, what really is interested in is looking at some of these inflammatory signals and how it associate with outcomes in AML. So in this plot here on the left, this is a pathway analysis from multiple, actually more than 700 AML patients, looking at their RNA, and we see a lot of correlation between a lot of the inflammatory pathways, including T-cell activation, inflammatory signals, interferon, immune activation, etc. What is also important is that these inflammatory signals are associated with outcomes, whereas patients with higher inflammation tend to have worse outcomes compared to patients with lower inflammatory signals. Yet we still do not know what are exactly these inflammatory signals that are at play, although we think from data from our lab, this is definitely based on AML T-cell interactions, which is why we took another approach where we looked at T-cells, particularly in AML bone marrows. This is a plot from single-cell data from around 24 newly diagnosed patients and 8 relapsed refractory AML patients. And each dot here represents a cell from CD8 T-cell. And then we try to ask simple questions like how do these cells coalesce between one group and the other? And what we see is that those cells that are coming from a relapsed refractory patients actually have distinct phenotype compared to T cells that are coming from newly diagnosed patients. What was also very pertinent is the biology of those T cells were actually different. So when we looked at the cytokine, or actually inferred cytokine in this case, because this is based on RNA data from single cell level, we see distinct cytokine or inferred cytokine differences between healthy and AML patients and also between the relapsed refractory and newly diagnosed T cells. Again, this is suggesting that the inflammation or the immune cell state of T cells is different, but this is really at the RNA level. What we also saw is there's interaction between those T cells and AML cells, which actually happen at a lot of the cytokines, suggesting that the AML cells and the T cells potentially causing some autokine or parakine interactions leading to stimulation of inflammatory signal within the bone marrow microenvironment, which could be systematic. So what we next did is we actually looked at the functional state of some of those T cells in the peripheral blood and in the bone marrow at the single cell level using cytokine profiling at that point. And that allowed us to understand that the dynamics of cytokine inflammation between the bone marrow and the blood is actually distinct. But again, at that point, we still do not know what are the key players that are leading to this inflammatory signal within the bone marrow microenvironment. I think this is quite relevant in our patient population who come to the hospital with acute illness in a sepsis-like picture, which really is represented by low blood pressure, sometimes oxygen requirements, fevers, but we do not find a clear source of infection. What we think these patients have in many cases actually on their imaging is leukemic infiltration into the lungs, inducing this hyperinflammatory phenotype related to ferritin as a marker of inflammation, ESR, and CRP. But again, this is still not clear to us if this is what cytokine are actually involved and what is exactly the players that are in that context. Because if we know the players, then we can actually treat them. Now, how is this related to what I just showed you in the prior few slides? We think this is all related to AML T-cell interactions leading to this inflammatory phenotype. But really what we were trying to look for is something that would allow us to investigate inflammation. And any approach to really try to do that requires to understand the cytokine. And there's a critical gap in that because we need something that is high throughput, that's able to detect multiple cytokines at a very fast rate with a low input volume, because these are patient samples. We're not always able to get high volume from the blood in order to test for the sera. We need something that is sensitive, meaning it's able to detect something at low levels, something that is reliable. We're able to actually repeat the data as well, and we're able to get data every time we test it, and something that can be used on historic samples. And what I mean by that is samples that are actually already stored and frozen from 5 years ago, 10 years ago, as part of banking of samples, where we can still detect cytokines in good quality. We did try different methodology, and we eventually also tested the NULISAseq, and this is some of the data, which is more of a pilot work that we've done on AML patients I'm going to share with you as a pilot experiment. So our sample description included 52 AML patients, 23 were newly diagnosed patients, and 29 were relapsed refractory. between males and female. There was 60% females in one group in the newly diagnosed and almost equal distribution in relapsed refractory patients as our pilot experiment. In this particular assay, the target was around 200 cytokines. Out of the 200 cytokines, we were able to detect 190 cytokines, and we didn't really see major differences in the cytokine profile in these 180 patients, in these 180 cytokines among the 52 patients between the newly diagnosed and the relapsed refractory samples. However, what really intrigued our attention is the correlation between the white cell number or white cell percentage in the blood and the sum of those cytokines. And this is relevant, although anecdotally, we tend to see that those patients that come ill with a sepsis-like picture tend to have high white count. Now, not all high white count patients do, however, do have that picture, but it tend to happen more. So this allows us to kind of get a window on some of those cytokines that could be involved in that context, as well as what potentially we can do to try to temporize those measures if we are to act on some of those cytokines. And this is just a correlation when we look at each of those cytokines, whether it is the same in the newly diagnosed refractory patients, it's actually similar, meaning the disease state itself does not matter. So this is more likely driven by the disease biology. And when you have high white count, you tend to have similar up or low cytokine profiling regarding those same cytokines. The other thing that we also see in the clinic is the relationship to monocytic leukemia. Monocytic leukemia is a subset of AML patients that have more of a monocytic differentiation, which are a bit more differentiated cells compared to the myeloid earlier progenitor cells. This is also relevant because monocytes and those cells could act like antigen-presenting cells, which means there are actually immune cells. And within those patients, we actually see a lot of what we call extramedullary disease, meaning the leukemia goes not just in the blood and the bone marrow, but tend to infiltrate other tissues, causing inflammation in the kidneys, inflammation in the lungs, and other organs, including the heart. And what we also see here is this tendency to have certain upregulation of certain cytokines compared to others. So this is a pattern that we're starting to see, and we're trying to dissect out which cytokines, which players are actually important. And the relevance of that is then we can go back and try to see if there are any inhibitors, which for many of those there are, that potentially could be used as supportive therapy in those patients. We actually also see some overlap between the cytokines depending on the clinical features of the patients. For example, if the blast is high and the white count is high, which can co-occur, we see some cytokines that actually go up together. If the lymphocyte is low, which is actually not uncommon for the lymphocyte to be low when the blast count is high, meaning the leukemia exists and the lymphocytes are actually lower, we see actually also some tendencies for certain cytokines to co-occur together. And this is relevant. I think this is more of a signal that those cytokines could actually also be coming from the leukemic cells and not necessarily only from the lymphocytes. So in conclusion, what we think is that NULISAseq 2000-plex inflammation panel was successfully able to capture high number of cytokines, more than 180 cytokines, which in our practice is actually significant and much higher than other tools that we actually tested in the lab, and also was definitely more reliable in getting some of the signals of certain cytokines we were specifically interested in. It is reliable. It actually was successful in all the samples that we tested. It is high throughput. There's definitely some differential association within certain subsets, specifically the monocytic. And from that, with the success of this pilot study, what we actually have done now is we're looking at a 250-plex cytokine profile from more than 400 newly diagnosed AML patients. And this data is still ongoing, and we're working on getting the data and then analyzing it and trying to identify patterns of response as well as features that could associate with certain cytokines and potentially improve patient outcomes. Thank you, and I'm happy to take any questions.
All right, wonderful. Well, thank you so much, Alex and Dr. Abbas, for your informative presentations. We will now start the live Q&A portion of the webinar. So if you have a question that you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. All right, so let's get started. We've got some great questions coming in from our audience here. And I know we have both of our speakers with us. I think we're having a little bit of some technical difficulties in regards to those cameras, but they are both with us here. All right, I'm going to go ahead and get our first question ready. Okay, so the first question asks, how is the 12 log dynamic range achieved?
Yeah, it's a great question. It's a big advantage for the NULISA platform. So it's actually showcased in the publication. So I definitely reference people to that for a detailed explanation. But we've actually created a patented approach where we can use a mixture of hot and cold antibodies to ensure that the signal for each protein is within the endogenous range. And so therefore, when you're actually measuring the protein with the sequencing readout, we essentially balance the sequencing reads to ensure that we can measure high abundant proteins with the same number of reads as we can with the low abundant proteins. So this means actually with NULISA that you can measure very, very small amounts of protein and high abundant proteins without the need for any kind of sample dilution by taking this tuning approach.
Wonderful, thank you, Alex. All right, another question here asks, is the CV comparison with Olink based on semi-quantitative measurements, and the CV is the variation of the signal?
Yes, again, thanks for the question. That's, again, is described in the bioarchive paper. So yeah, for the multiplex panels, right now we have relative quantification based on the read count from the sequencing readout. So the CV that comes from that is essentially variation of the signal that we see for each assay within the sequencing readout. What we do see though is CVs, equivalent CVs of less than 10% for the singleplex assays with the qPCR readout, which is absolute quantities, as well as the multiplex readouts. So really the CVs are very consistent, whether you're measuring large numbers of proteins or single proteins, typically less than 10%.
Great, thank you. Another question we have here, are there any other application areas that would be suitable for this panel or technology?
I can take the first stab at that. Dr. Abbas might have some specific thoughts on that with regards to leukemia and the work that he's doing. I think there are many applications for liquid biopsy proteomics in the cancer field. I mean, early detection and diagnosis is obviously one of the key ones that I highlighted. But here, I think protein biomarkers can be associated with different cancer types and early stage disease. It's going to play a huge role. I think cancer subtyping, so looking at proteomic signatures from liquid samples to enable understanding of severity of disease or which type of cancer is presenting itself is another key area, as well as monitoring treatment response. And another hot area, actually, where I think proteomics can have a big impact is probably minimal residual disease, MRD. I think this refers to a small number of cancer cells that remain in the body after treatment. And I think proteomics can actually play a key role in monitoring that as well. And I think stratifying patients for clinical trials or predicting drug response is another area where proteomics in cancer is going to play a key role, and where sensitivity is going to be very important. I don't know whether Dr. Abbas has any other thoughts on some of the work that he's doing and how this might apply there.
Yeah, Alex, thank you. Yeah, in addition to what you mentioned, I think there's also the application of treatment toxicity, especially in the era of immune-based therapies, especially when it comes to CAR-Ts, checkpoint blockade, all of those. Treatment with anti-inflammatory drugs and monoclonal antibodies is mainstay, but sometimes we try to go, with our best guess or use something like big gun, such as like steroids or cytoxin or cyclosporins to kind of cave autoimmunity, just because we don't know exactly what's the molecule that is really playing the role there. So I think having toxicity and understanding disease pathogenesis and also building treatments based on that is definitely critical. There are many cytokines now that already have drugs for them, and just knowing when to use them is going to be key. In AML, as I mentioned, there's also this category of those patients who have this extramedullary involvement as well as infiltration of the leukemia in their tissues causing inflammation. But we really don't know what exactly is the element. There's some data about IL-6, but beyond that, there's not much. I like the idea of MRD and trying to look, because this is a big thing in AML. We know in leukemias, MRD is a major prognostic factor. And inflammation in the last 4 or 5 years, and even maybe 10 years, but particularly the last 4 or 5 years, with the role of MDS, AML, leukemia, even CHIP2 leukemia has been emerging and kind of been emphasized. So trying to understand more some of those players is an area of a lot of interest to me as well, something that we're hoping to pursue with this.
Yeah.
Great, thank you. We have some more questions coming in here and a little bit more time so we can continue. This next question asks, have the NULISA validated in neurobiomarkers other than PT217? Do you have a multiplex panel on these neurobiomarkers?
That's a great question. Actually, we do, and we're really excited about the panel that we have. So first of all, we've actually validated a number of important neurodegenerative disease biomarkers, including p-tau217, p-tau181, p-tau231, NFL, GFAP, and amyloid beta 42 and 40. And these are really the critical biomarkers in Alzheimer's disease and other neurodegenerative diseases. We also have a very sensitive assay for alpha-synuclein, which is critical for Parkinson's disease, for example. We didn't include it in this presentation, but we do have a CNS disease panel with 120 biomarkers, which can all be measured with very high detectability from plasma or CSF. And that incorporates all of these neurodegenerative disease markers and also the inflammatory markers. So you can really get a very broad view of sort of neuroinflammatory changes occurring in patient samples.
Great, thank you. Our next question here asks, is NULISA more challenging for antibodies with high cough?
I'm not sure what high cough is, so I can't really answer that. All I will say is that we spend a lot of time screening for the best antibody pairs for NULISA, and we typically use monoclonal antibodies from commercial sources. So typically we find a good antibody pair and then validate the assays, and that's a fairly high throughput process now and fairly quick to develop new assays. So I'm not quite sure whether I specifically answered that question, but typically we can find commercial antibodies that work well with NULISA. If not, we do have a secondary technology at Alamar called antibodies, which we occasionally utilize to develop ultrasensitive NULISA assays as well.
Great, thank you. And to that attendee, if you'd like to submit another question for additional clarity, please do. We're happy to answer that. All right, moving forward, this next question here asks, what causes the inflammatory phenotype in AML, the normal immune response, or rather the interleukins secreted by AML cells themselves? Could NULISA help discern between these?
Yeah, this is a great question. This is actually a major area of the lab now. We do have actually a study under revision now discussing that. I think there's actually an interplay between the immune system and the AML cells. As a reminder, AML cells are monocytic or myeloid cells, which are immune cells that could make inflammatory markers by themselves, but also could induce T cells, could induce some distant effects, etc. So there's an interplay. It is likely both. And can NULISA answer that? I guess we'll find out. I do think that some of the data we've received so far, what I showed today was really pilot, but it just tells us that we can detect a lot of cytokines try to go after. And in fact, we're proposing to do more of those by co-culture assays from in vitro samples as well, and then apply it on NULISA. So I don't know the answer yet, particularly which is the chicken or the egg, because this is really the question you're trying to get to. But I think so far we know there's an interplay, and both the immune system and the AML cells themselves could induce some of these markers.
Great, thank you. Now our next question here asks, this question is for Alex, and this question asks, Alex, can you speak to any planned target expansion of the current panels or plans for adding new target analytes, particularly in the oncology or immuno-oncology space?
Yeah, that's a great question. Thank you. And yeah, we are planning a couple of developments in the next few months, specifically in the oncology space. One is actually going to be a mouse panel, which will enable high sensitivity detection of oncology and inflammatory markers in mouse samples, obviously for preclinical work and mouse model work. And then we're collaborating with a number of partners to identify oncology specific content that we can build into a sort of immuno-oncology based panel, and also looking for early detection biomarkers that we can create into a panel. So I think our overall strategy around content is to identify critical disease specific markers and put them into cost effective and highly sensitive panels. And oncology is certainly a major focus there, alongside neurology and inflammation.
Great. Well, thank you so much, Alex and Dr. Abbas, for your time today and your important research. This does conclude our session, but before we go, I'd like to thank the audience for joining us today. And I'd also like to thank our sponsor, Alamar Biosciences, for sponsoring today's webinar. Questions submitted during the talk and the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until October 7th, 2025. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.